Hi, everyone. Good morning, good afternoon, or good evening, depending on where you're joining us from. And on behalf of the Society of Neuroscientists of Africa, SONA, it is my pleasure to welcome you to another edition of our SONA webinar series of 2020. So, whether you're joining from Africa or elsewhere, um, thank you so much for choosing to spend time with us. Each month, you perhaps are aware by now that this series continues to bring together students, clinicians, researchers, educators, and neuroscience enthusiasts from across the world for conversations that advance neuroscience on our continent. So I am Dr. Royhaan Folarin. I'm an assistant professor of human anatomy at the University of Global Health Equity in Rwanda. And I also serve as the treasurer of SONA, as well as the coordinator of the SONA webinar series. So it's therefore my privilege to serve as your moderator today. And for those joining us or joining this webinar for the first time, SONA stands for the Society of Neuroscientists of Africa. It is the premier continental organization that is dedicated to advancing neuroscience research, education, collaboration, and capacity development across Africa. We have our membership spanning through every career stage. Whether you're an undergraduate student or you're a senior investigator, we represent institutions throughout the continent and beyond. And our vision is simple. It is just to strengthen African neuroscience through scientific excellence, through collaboration, through mentorship. And the webinar series is one of the ways that SONA pursues this mission. Our goal is not merely to host lectures where we just talk. Rather, we aim to create a community where African neuroscience is showcased, where neuroscience is discussed and continually strengthened. So accordingly, the webinar series intentionally alternates between presentations of different themes, from scientific to career development sessions, and thereby exposing participants to cutting-edge discoveries that is going on within Africa and to experiences that shape successful scientific careers. So as the series grows, we continue to remain committed to representing scientists from across regions, Northern Africa, Southern, Eastern, Western, and across neuroscience disciplines, across institutions, across career stages. So Africa has extraordinary neuroscience to offer. If you agree, that's what we're saying. And this platform exists to make those stories more visible. So today's webinar is particularly exciting because it illustrates fundamental molecular neuroscience and how it can be translated into population-level impact beyond things you just read in the books. So dementia is rapidly becoming one of the most important global health challenges. And despite this, Africa remains underrepresented in many of the studies that shape our understanding of these diseases. So I won't preempt the presentation further than that, but you need to know that today's presentation speaks directly to that gap. So before we begin, I just have a brief announcement before I talk about our speaker, and that's the fact that at the conclusion of the webinar, we always share a feedback form, which is extremely valuable to us because it guides improvements to future webinars and it helps us identify topics of great interest to the neuroscience community. If you wish to receive a certificate of participation, you definitely have it after you fill in you give us your feedback. You tell us what you think about the webinar. So throughout the presentation as well, please feel free to submit your questions using the chat, and we'll reserve the time after the presentation for discussion. So it is now my distinct pleasure to introduce our speaker. I mean Professor Mahmoud Bukar Maina, who is a British-Nigerian neuroscientist whose work has become widely recognized both within Africa and internationally. How are you doing, Prof?
I'm doing all right. Thank you for having me.
Alhamdulillah. So, Prof. Mahmoud serves as Wellcome Trust Career Development Fellow. He is based at the Biomedical Science Research and Training Center, BioRTC, which is domiciled in Yobe State University, Nigeria, where he is the director. And he also holds appointment with Sussex Neuroscience at the University of Sussex in the United Kingdom. He's a professor of neuroscience at Newgate University in Nigeria. And like I said, he directs the BioRTC. So I'm sure you read the rest of his bio in the flyer. If you haven't, you can read more, like we included there. So his research, which is why we're having him here today, explores the molecular mechanism that is underlying Alzheimer's disease and related tauopathies, with particular emphasis on amyloid beta toxicity, amongst others. He's here to benefit us with how much his research and his team has been able to contribute and are contributing to the African and international database. He recognizes the urgent need for African-led neuroscience infrastructure, and that's why he established the BioRTC to develop open access iPSC biobanks, dementia cohorts, amongst others. And beyond laboratory, he has played major roles in scientific capacity building policy, as well as mentorship and an open science initiative across Africa. Prof. Mahmoud will be speaking to us today on a topic titled From Molecules to Population, Building an African Platform to Understand Dementia. And the presentation, like you can imagine, promises to take us on a fascinating journey from molecular mechanism to translational neuroscience, amongst others. So without further ado, and having said quite enough from this end, Prof. Mahmoud, please join me. And I encourage you, all our listeners, to also welcome Professor Mahmoud Bukar Maina as he takes the stage away. The floor is yours.
Thank you very much, Dr. Royhaan, for the introduction and for the invitation to you and the entire team. I hope that I am audible. I hope that you can hear me. If you can, then I can now continue my presentation. Am I audible enough? Can you hear me well? Can you hear me, Dr. Royhaan?
Sure, I can. You have the floor, loud and clear.
Oh, fantastic! All right, thank you very much once again.
Yeah.
So what I want to do is to try to walk you through my journey by telling you a little bit about myself and how I kind of stumbled or how I kind of got into dementia research and what we are doing now to try to help us understand dementia from across diverse ancestries. And I think this is very, very important for this audience simply because, I am an African and my journey might resonate with a lot of people. So it's important for you to see how I started and where I'm heading or where I have come so far. So, of course, I was born and raised in Yobe State in Nigeria, specifically in Guru. And I went to my primary school at Nguru. And then following that, I went to my secondary school, FGC Bunyadi in Yobe. And that's actually me with my classmates. I think this was probably in SS1 or SS2. And then following that, I went to Borno State to the University of Maiduguri, where I was med. They say if you want to be med, come to UniMed. So I did my bachelor's degree there in human anatomy. When I finished that, I did my service at the Federal Neuropsychiatric Hospital Maiduguri, where I started working before moving to Gombe State University, start working in 2011. So really, my interest in neuroscience started quite young when I was little, and then it became really shaped by my experience during undergraduate degree and consolidated to some extent when I was at the Federal Neuropsychiatric Hospital Maiduguri. But one thing that really kind of caught my attention across these different places is the fact that we do have resource challenges across these different places. It could be equipment, it could be the fact that we don't have as many mentors, we don't have as many opportunities. And that kind of fuels science misconceptions that continues to be, prevalent across different communities. And then, of course, this influence the policy or kind of creates a gap, and this creates a positive feedback where you have a problem, resource challenges, problem that kind of perhaps enhance science misconceptions, or the other way around, on that kind of like continues this loop. And I tried to really understand this more deeply when I started, building myself towards having my own group, by trying to understand what is the major challenge affecting African neuroscience, for example. Is it really true that that suspicion about this lack of challenge, or resources and other things really shapes the neuroscience ecosystem in Africa? And I had a real reason for doing that. But yes, we looked into neuroscience across the continent, and what you see here is that across the continent, the use of advanced techniques here could be Western blotting, could be qPCR, in neuroscience laboratories across the continent is very, very low if you compare it to other places. So that really kind of course, influenced me to migrate. Some people, in Nigeria like to call that Japa. I'm not, I think, I don't know if Japa is from Yoruba or something, but it means that you leave your country to another, to a different place. So that really made me leave Nigeria to go to the United Kingdom to pursue my ambition about neuroscience. And of course, I obtained my PhD in 2017. In 2022, I secured a competitive funding from the Alzheimer's Association that allowed me to start up my group, independent group at the University of Sussex. Throughout this journey, some of the highlights about what I've been able to do is understanding how amyloid beta and tau influence Alzheimer's disease development. And we use diverse techniques in the lab, from imaging to genomics to biophysics, and now even including clinical evaluations. So this is really me. But before going into how this journey of mine from childhood to what we are doing right now, how or why it matters, I think we need to ask this question: why does representation matters really in science? And I'd like to show you this. So this is the world as it is today. Clearly, everything looks normal. Africa looking great, big compared to some other places. Only that if you use the same website where this map comes from, which is the World Mapper, to look at Africa by epidemics, you see that Africa is well, huge or well or represented. And then if you ask another serious question, how about deaths from epidemics? Unfortunately, you see that Africa again is like the entire world compared to other places. So you'll be thinking that if this is the case, we should have more science coming from Africa to help us understand these challenges. But unfortunately, if you look at the world map by scientific publications, you see that Africa barely exists. Now, this clearly limits African populations from fully accessing global biomedical advances. But then, if we look at the world as it is, we know that we are not all the same. Taking example of, with Nigeria, I am from Yobe State. Even from Yobe State, there are many different ethnicities. Half of me is Fulani, half of me is Kanuri, but there are many different ethnic groups within Yobe. And if you now look at Nigeria as a nation, it's so huge that you see someone and you might not even understand if the person is from Nigeria. I mean, a lot of people from the South think I'm from Ethiopia, or I'm from, Rwanda, or I'm from Uganda sometimes. So really, this tells you that humans are diverse. And this diversity matters when we talk about science, especially if you think about this biologically or genetically. Africa has the highest genetic diversity of all continents. So this is based on 1,000 Genome Project showing how diverse the world is. And Africa, you can see, has the highest genetic diversity of all continents. And this genetic diversity means more potential to discover protective and risk variants for different diseases, not just for Alzheimer's disease or dementia in general. And I'd like to give you a case study of why this diversity matters, specifically with dementia. But before going into that, this is the worrying part. Dementia in the next 24 years is expected to increase in Africa by more than 300%, depending on region. But Nigeria, right there where I was born and raised, is going to see over 360% increase in dementia. As the rest of the world is calming down dementia, Africa is going to see this. So this is really a major problem. But then one of the greatest genetic risk factor for Alzheimer's disease development, and Alzheimer's disease is the greatest cause of dementia. So one of the greatest genetic risk factor for Alzheimer's disease development is this ApoE4 allele. And so ApoE4 or ApoE is a protein that comes in different versions or flavors. I like to call this the good, the neutral, and the bad. The ApoE4 is well overrepresented in Africa. So more Africans have more ApoE4. Paradoxically, if you look at ApoE4's influence on Alzheimer's disease, you see that there's a clear ancestry influence, such that if someone is a Japanese, the influence of ApoE4 on Alzheimer's disease development in them is more than if they're Europeans. And the more Africans one is, the less they are kind of influenced by ApoE4. Even though I like to tell people that when we say Africa, Africa is heterogeneous. And the data giving us this information, I don't think is complete because we haven't really studied diverse Africans. But for now, let's say that ApoE4, this is what it does. So the reality is that if we do not study diverse populations, we miss fundamental biology and the pathways to effective therapy. But to do this, we need cohorts matched with cellular models. And throughout history, a history of dementia over the last 100 years, we know that well-characterized cohorts have really transformed mechanistic understanding of dementia, specifically, let's say, Alzheimer's disease. And I would like to give you a short story as a digression. And I'm going to take you to Colombia specifically, Medellin. This is Aliria Rosa, and these are her two children. So Aliria belongs to a cohort that is called the PISA AD cohort or the PISA cohort. They are this cohort, there are thousands of people from this family, and over 600 have this mutation that causes them to develop an early form of Alzheimer's disease due to mutation in presenilin. So if you have that mutation, based on the scientific evidence, you are guaranteed to develop an early form of Alzheimer's disease. But Aliria lived up to the age of 73, I think, before she passed away, and she never developed Alzheimer's disease. So that was baffling. How could she have a mutation that should have caused her to develop dementia early on, but she didn't? And it turns out that because Aliria and many people from her family are part of this cohort that has been well studied, using fluid biomarker, using genetics, using many different approaches, it turns out that she has a mutation in her ApoE region, specifically ApoE Christchurch mutation. And it is this mutation that kind of overrided the other mutation, the effect of the other mutation. And this really was a breakthrough in the field like 4 or 5 years ago when the paper was published. And there are a lot of studies now trying to produce ApoE4 Christchurch mutation mimetic drugs that can be used towards protection. So the lesson here is that this is a discovery made in Colombia, far, far away from Europe. And it's because they have access to the cohort, people with their fluid biomarker, genetics, and many other things that allow them to really understand this. And this is exactly one of the ways or one of the powers of well-characterized cohorts. But the question is, do we have similar dementia research platforms in Africa? And this is really important because if you look at dementia research globally now, you see that obviously it's really high. But then if you look at Africa, you see that it's like a flat line, which is a major challenge, given that in the next 24 years, we are going to experience this major increase in dementia cases. And again, also, if you look at the genome-wide association studies conducted to date, not just from 2012 to 2022, you see that indigenous Africans are not included, or they are lacking. This is amount of GWAS data coming from Europeans, East Asians, African Americans or Caribbeans, and this is from multi-ancestry groups. So basically, again, Africa is missing in action. So the reality is that a risk map for Alzheimer's disease, for example, that is drawn from one ancestry is not a human risk map. And these missed ancestries represent a missed mechanism, because we don't know what we can discover, similar to what has been discovered based on the case of Aluria, if these different ancestries are included. And another thing to mention is that Alzheimer's disease etiology is not the same across populations. So, for example, this is a popular kind of figure used by scientists when presenting about the evolution of Alzheimer's disease over time, showing protective factors and risk factors. So it's kind of like a continuum, a battle between these two factors. But I cycle education and genetics because education is known to increase cognitive reserve, which lowers or which is protective against dementia. But then again, let's take Nigeria as an example. Even within Yobe, there are different forms of education. There are those that go to entirely, Islamic type of, non kind of structured education, and there are people that go to that. So even in the education setting, there is a lot of variation across not just Yobe, but across Nigeria and Africa, that we don't really know how that influences this, and therefore a lot of the data is based on education using Western form of or Western style of education. So that also means we don't really know much about the etiology of Alzheimer's in our own population. And genetics, I just finished mentioning how genetic diversity influences dementia development. So, and again, also if you look at the risk factors, smoking, hypertension, diabetes, alcohol misuse. So there are a lot of different things also that are different, some are similar across Africa and with other ancestries, which kind of means we really have to study our own population to fully understand how dementia starts and progresses. So the reality is this is a biological problem, not an equity problem, because some people think that when we talk about this, it's about equity. It's about wanting Africa to be represented. But actually, it's not about that. It's about biological discovery problem. Perhaps Africa is the missing piece of puzzle to help us understand and defeat dementia on a global scale. And this is where I come in. So in the Maina Lab, our scientific mission is to harness African ancestral genetic diversity to uncover the molecular mechanisms underlying dementia. And where we are, or where are we? So we are based at two locations. Most of my time now is in iPSC in Yobe State because I recently relocated back to Nigeria after 15 years of my time in England. So most of my time is in iPSC now, and I still maintain a lab at the University of Sussex. So therefore, the work that we do is kind of some in Nigeria, some in the United Kingdom. And the scientific approach that we take is to go from cohorts to mechanisms. Because I was trained as an anatomist, I did my master's in cellular and molecular neuroscience, PhD in neuroscience, postdoc in biochemistry, and then I started developing interest in cohorts. So basically, all this experience made me understand that if we really want to understand dementia, I believe we need to go from humans, from the clinical side down to the bench. And so this is the approach that we take in the lab. And I'm going to start by telling you stories about what we are doing at the cohort level, then take it down to the modules and mechanisms. Now, we started by establishing this Sahel study, which is study of ancestry, health, environment, and late life neurodegeneration. And it's largely focused in northern Nigeria, but we also have some kind of contacts with the neighboring region because some of the hospitals that we have there, we do see people coming from Chad, Niger, and Cameroon. And this was when it was launched, with different hospitals funded by Alzheimer's Association and Rainwater Charitable Foundation. And we have about 6 different hospitals that are part of this. And one of the evidence that really motivated us to do this is we start by evaluating dementia cases in the region, by looking at kind of hospital data sitting there to understand what has been happening over the last 20 or 30 years. And this led us to publish this work, which showed us that in the region, we have a lot of the Alzheimer's type of dementia, but also vascular dementia. And we could also even categorize, the social class of the individuals that are, that are affected, from laborers to those that are really kind of doing well. And then we also have diverse, as ethnicities, Kanuri, Fulani, Shua Arab, etc., etc. So that paper showed us that over the last 20 years, in one facility, Federal Neuropsychiatric Hospital, for example, over 1,200 dementia cases have been reported. And this is a large untapped dementia population that is sitting there in the Sahel, a region that has never been studied, because northern Nigeria is a region that is a bit pervasive to medical research because of historical mistrust from scientists coming to take advantage of the population. So this is something, or a population, that is unique in a way. So for this study, we have a scientific advisory board that is comprised of geneticists down to ethicists that guide what we're trying to do. And so the study design is basically to go from first building a cohort from the community and also from the hospital that will now be diagnosed, used, or characterized using diverse batteries, from neuropsychological batteries to imaging, fluid biomarker, and GWAS. And from this, we have therefore the hospital cohort and we have the community cohort. So from the hospital cohort, this is based on the data from the last, I think, 4 months, because this data has now that has been updated, which is not included in this. But basically, these are people from the one of the hospital that have dementia, and the age, male and female, and the age group. And these are some of the batteries that we use to evaluate them. So in my team, we have neuropsychiatrists, we have nurses, we have lab scientists, and of course we have basic scientists, etc. So this is the work of the collaborators in the hospital. They use MOCA, QDRS, NPIQ, so different batteries to really kind of clinically characterize this population. And of course also we have the community cohort. So this started last year, where we did a massive community exercise that allowed us to recruit over 1,200 people. We did a lot of things, using Minicog, using many different things to evaluate at the community level people that may be at risk of dementia and those that have probable dementia. And this is also the age range, the ethnicities represented, the level of education. And as I was mentioning earlier, as you can see, the level of education from this community cohort is largely Quranic education. So this is really, really important, I think, because it means that our cohort has a uniqueness that many cohorts don't have, because of this, the fact that most of the people do not even go to formal or Western type of education. And again, also, these are coming from different ethnicities. We have about 8 or more different ethnicities represented. Again, we did evaluate this using different batteries. So what we are now doing is to now commence fluid biomarker analysis, which is done in collaboration with Thomas Karikari using the NULISA platform. And then, of course, we are also using brain scan. This is in collaboration with Oduna, a professor at McGill, and Chadi is the clinical lead of this arm of the work. And of course, we are also sending samples for genetic analysis. And importantly, since last year, we started collaborating with Professor Odeo Momo, who is at Wake Forest University, but also at Aga Khan University in Kenya, who is leading the Africa Fingers project. So I am leading the Northern Nigerian Africa Fingers project, but basically this is also complementing what we are doing, because it's a project that wants to identify people that are at risk of dementia, so that lifestyle intervention can be given to see if that can slow their progression to dementia. But the reality is that building cohorts alone is insufficient unless they can be connected to experimentally tractable systems capable of interrogating mechanisms. Because think about it, if you have human that you understand their fluid biomarker, their genetics, their cognitive function, but then you move to mice model to now try to understand their behavior, and then you move to another model to understand at cellular level. While that is good, but it won't really give you a match of the reality. So if you have the ideal system is you have the human, you do those evaluations, then from the same human, if you can build a model that you can now take it to the mechanism. And this is exactly what we're trying to do. So this now brings me to the second part of the uh, talk, which is building models from the same cohort. And to do this, we are using the iPSC modeling. So iPSCs stand for induced pluripotent stem cells. And basically, for those that have done embryology in their undergraduate education, you know that after the male and female meet, the zygote forms, and then that leads to the formation of the blastocyst. And within the blastocyst, we have embryonic stem cells. And it is these embryonic stem cells that now end up going to develop cells of the ectoderm, mesoderm, or endoderm that makes us who we are as humans. But then this process is ethically kind of very challenging. You can't really get access to embryonic stem cells easily. But then in 2007, Yamanaka and colleagues showed that you can actually be able to transform somatic cells like fibroblasts, like blood cells, like hair cells, like urine cells. You can actually be able to program them into iPSCs. And these iPSCs have similar properties. So in theory, you could say that iPSCs can also be, grown into a human being. Of course, that is not acceptable. But the idea is that with the iPSCs, we can now be able to produce cells of ectodermal origin, such as brain cells, to now understand from the same individuals, collect skin biopsies, that we can now be able to grow brain cells from the same individuals to understand disease mechanism. So this is a paper being prepared by my research assistant, where unfortunately it shows that iPSC resources, despite Africa's huge genetic diversity, are absent from global resource. These are iPSC biobanks, not the entire ones in the world, but those that have met certain criteria. But Africa, none is existent. So this is really, really sad and very unfortunate. So because of that, therefore, one of the things that we are doing as part of this work is to establish an open access iPSC bank. In fact, we've already established the first African iPSC biobank that is open access, that is accessible to the global scientific community. And so what we are doing now is, from the same cohort, we are collecting skin biopsies or blood cells for those that find skin biopsy to be invasive. And now we are generating iPSCs. Some of this we are converting to ApoE lines that we will use to understand the role of ApoE in dementia. So this is just to show you, for example, how we do this. When I started, I was at the University of Maiduguri. This is one of my clinical collaborators, Professor. One of my clinical collaborators, and he collected this skin biopsy, which I now put into a tube with media, and then I take that media. At that time, I was using the lab of my mentor, Professor Issa Hussein Marte, to now process that into fibroblasts at the University of Maiduguri. So basically, I process the biopsies. This is a biopsy. These are fibroblasts that grew from the biopsies, and we could confirm that using different techniques that these are the fibroblasts. But unfortunately, at that time, there wasn't enough resource, like the equipment and other things, to do this locally, to convert this into iPSCs, locally. So I had to take the cells to my lab in Sussex, where my two students at that time, Zaid, who is currently doing his PhD with me, and Phoebe, who is currently doing her PhD at the University of Newcastle, worked on generating iPSCs from this, and they could confirm that using different techniques. So basically, to the best of my knowledge, this is the first iPSC line from an indigenous Nigerian, and one of the first, if not the first, published from an African. But I know that there are other groups working on iPSCs in South Africa, which is really, really fantastic. So I think this is going to be an area where we can all work together to see the expansion of this in the continent. So really this inspired me to establish the African iPSC initiative, which is basically how can we collect as many iPSC lines that we can bank and make it accessible to the global scientific community. And therefore, to start that process, I now said, okay, why don't we collect more biopsies from diverse ethnicities, in Yobe for a start, that we can now generate iPSC lines. And this is the work that we've been doing in collaboration with Celesta Catch. one of my mentors, a fantastic scientist at the at Washington State University in St. Louis. Basically, we collected biopsies that we converted to fibroblasts that we now converted to iPSCs. We could confirm they have normal karyotype using karyotyping. So basically, this is a paper that has been published, and this is one of the team. This is a team responsible for biopsy collection. This is the plastic surgeon in a team, and this was my postdoc at that time, Dr. Murtala, who is currently in China, but he's coming back to us hopefully in September. And this was my PhD student, Abdurrahman, who recently completed his PhD, working on generating iPSC lines from centenarians. But what we have done is we've characterized this, genetically by doing GWAS and RNA sequencing. For example, it shows that these lines are aligned, or they are of African origin based on kind of using reference from the 1000 Genome Project. So they are far away from other lines from either Europeans and other places. And we could now do genotyping of ApoE. It shows that a lot of them have E3, E3, and some of them have E2, E3, and some of them E2, E4. And we've also done polygenic risk score calculation, showing that, for example, if you include ApoE, they largely show similar risk profile. But then if you remove ApoE, you see a little bit difference in comparison to the reference population. And then if you do risk calculation for Parkinson's disease, you see that it's a little bit different. And if you now try to do risk score calculation by ethnicity, you see that there are kind of major differences coming up. Although I should say that this is really preliminary, this is on a small sample size, and polygenic risk score that we know of today are largely based on genetic data from European ancestry population. So the model can probably not be able to give us an accurate polygenic risk score for Africans. But the reality is that the fact that we are seeing some differences emerging by ethnicity means that this is going to be really, really important as we go forward, especially in our cohort that is multi-ethnic. So what we've done is to generate iPSC lines from that have MAPT mutation to help us understand tauopathies, because this is something that is of interest in my lab, but I'm not talking about this in today's presentation. And all these lines are available to the scientific community globally to access them and use it for their research. So I've talked to you about cohorts and models, and now I'm going to try to quickly, hopefully in the next 10 minutes, summarize everything and talk to you about mechanisms. Because what we want to do is to say today we have person X. He has a risk profile that may mean that he may develop Alzheimer's in the next number of years. And we collect all that information about their fluid biomarker, genetics, MRI, and other things, and then build model from the same individual that can allow us to now culture their brain cells in a dish that can help us understand, even before they develop that, how it's going to look like. And this is therefore the approach that will help us towards personalized medicine. So this is why from models we now go to mechanisms. So what we are trying to do is to now build, for a start, astrocytes and neurons or co-culture and eventually brain organoids that we can now apply these different techniques like RNA sequencing, mass spec, lipidomics, and others to help us really understand how different ancestries, because in the lab we grow cells from European background and from African background, so that we can understand how different ancestries influence disease development. And of course, eventually what we all like to do in the lab is to be able to now say, we have cells from Africans, from different ethnicities, from Europeans, from from Arabians, from these. I believe that this is the approach that can allow us to truly have a global understanding of dementia, to have an effective therapy towards it. But one thing I want to highlight is this work that's currently being done by my postdoc, Dr. Suleiman Koiranga, who is really, really talented. What he's done is to try to say, okay, what if we mine, existing genetic data, can that reveal some variants that are missing, that are of African origin, that may influence disease development? So he applied this 6 bioinformatic pipeline that allowed him to kind of identify 12 Africa-enriched MinSES variants that are never tested in any Alzheimer's disease GWAS, and 6 of them are associated with either tau propagation, amyloid, or kind of inflammatory pathways. So one of these candidates is the bin kind of variant, which is by a tau by which is on the tau binding domain and is carried by about 9.7% of Africans, as compared to 0.02% of Europeans. What is really, really interesting about this is that the Professor Hinyemi's group in Ibadan, they recently kind of published one of the abstracts from their work showing that this same variant is protective against Alzheimer's disease. So this really shows the power of this kind of bioinformatic approach to try to dig in to see, based on the existing data, what is out there. Is there something that we can discover that we don't know of right now? So what Suleiman is going to try to do now is to kind of screen our cohort to see if they can identify this particular variant, and then build iPSC lines from this same variant, people with this variant, to now generate iPSC lines that can allow him to understand how this influences our biology. So I believe this is perhaps the platform that would allow you to make discoveries that will be matched or similar to what we see with the Colombian example. So this is the platform that we have right now, the Sahel platform. We have a cohort of over 1,200 people. We have at the moment, I think, over 100 that have clinically confirmed dementia. And we also have a cohort of chronic kidney disease that is that we can easily tap into, that has over 2,000 people. And these are some of the phenotyping that we are doing. And we also have over 1,200 plasma, serum, and bufficus from the same cohort, over 900 skin biopsies from the same cohort that can allow us to kind of go back if we identify something to say, let's get the biopsy from individual X that we can now use to generate iPSCs or to generate some other cell types. And already we have about 11 iPSC lines generated. So really, this is the discovery platform that I was kind of alluding to at the beginning of my talk, that we need to build to help us make groundbreaking discoveries, that is driven from within Africa. So perhaps ApoE research was discovered through investments in a deeply characterized population, and of course that's the case. Our goal is to build this infrastructure needed to uncover many more such discoveries in African populations. So in the next 5-6 minutes, I know we have to give time for questions. I'm going to try to now shift gears, because most of what I've talked about are things that has been done largely in Europe, with some of them done in Nigeria. And I truly believe that for us to advance scientific understanding, we need to build a local capacity that will take ownership, that will build local trust to now do this level of science. I am a title holder in the Emirate of the Matru, and I know how people get skeptical by someone coming to the community to say they want to collect samples from them. That is something that does not create sustainable science. And so in my own case, most of what we do is we send samples to my lab in Sussex or to USA for analysis. While we want to continue doing some of that, but what we want to do, or ideal scenario, is to be able to do large chunk of that locally, and for the control of that data to be driven locally as well. I've already shown you, however, that in Africa, we have this challenge about the infrastructure. And this is exactly what led me to establish the Biomedical Science Research and Training Center about 5 years ago in Yobe State, at the heart of the Sahel. The interesting thing is that Yobe State, the motto of Yobe State is the pride of the Sahel. So we have a Sahel cohort, and we have BioRTC located at the heart of the Sahel. So this is BioRTC, our new building that we moved into in January. We have even a sport facility so that scientists doing their science can have a fun time. We have about 50 kVA solar that allows us to be constantly with electricity. And of course, which is sad, but we have some of the only technologies in Nigeria. For example, our center was the first to have a confocal microscope in Nigeria. Now we have 2. So we do have like fantastic, equipment in this center, which I believe will now allow us to take the local ownership of doing this level of science that I'm doing. And in fact, this is one of the reasons why I migrated back to Nigeria, to ensure and to show to the world that we can do this level of science locally. And already locally, we are able to now start generating somatic cells. This is work being done by my research assistant Dawood and my PhD student as of that time, Rahman, both from fibroblasts and from PBMCs. And these are iPSC cells generated from indigenous African, and I believe this is probably the first indigenous African centenarian iPSC line generated. And so all these lines, if one wants to have access to them, all they have to do is to go to the BioRTC website, go to the resource, go to the African iPSC initiative, and then within there, they can now go to the cellular bank, and there they can now see all the lines that we have and the information around them and how to access them. Of course, we charge for access, not for profit, but if you're an African scientist working on this, if you don't have funding, let's talk. We will see how we can give you cells so that you can use them for your work. And in addition to that, we are committed to building capacity, because this is the only way that we can build sustainable ecosystem. We need more people that can do this level of science locally, so that they don't have to go to Europe or America because of the lack of the equipment. If they want to go, let them go, because they want to just experience another culture, which is what most Europeans do. You see someone coming from Spain to England to do science, not because the equipment is lacking in Spain, but just because they want to experience a new environment. This is what we want to build in BioRTC. So since founding it, we've trained over 1,000 people from across Africa. And we also have a very good relationship with the community and with the government. And I'm an advisor to the Yobe State Governor for Science, Research, and Innovation. So that really has helped BioRTC to kind of be embedded and be kind of trusted by the community. So our vision in overall, or in summary, is to go from human diversity to molecular mechanisms in Alzheimer's disease and related tauopathies. What we want to do with the cohort that we have and we are building is to stratify genetic risks and resilience for dementia, for example, that we can now take to model this within kind of a model system, using iPSCs or using directly induced neurons, that we can now take to the next level to determine how genetic variations shape phenotypes across ancestries. And we want to make everything that we produce open access. Of course, the control of the data has to be locally driven, but we want people to access this resource. So I would like to really end by acknowledging a lot of individuals, collaborators, funders, and people, and as well as participants and patients that are part of our work that we do. I'll leave you with this slide. Alzheimer's disease will not be solved by studying a small fraction of humanity. If we want to understand the biology of dementia, we must study the full diversity of the human species, and Africa is central to that. Thank you very much.
And we also thank you very much, Professor Mahmoud. If we had some water here, we really could pass over. That was a very fantastic presentation, as always. I hope you can hear me clearly. Thank you so much. That is highly appreciated. Indeed, of course, I've listened to your talk a number of times, but it's always refreshing how much your commitment to advancing the African community and indeed contributing to international excellence from the African platform. It's always been a very inspiring thing to listen over and over again. So you have reminded us how many firsts that you have recorded from the heart of the Sahel. Thank you so much. That's impressive. And now, our dear audience, is the time where we get to take your questions. We have had a very robust engagement from across Africa and also beyond. Some of them we managed to show on the screen and others are not. But please, in case you still have questions, please drop them. And I'm going to start with the questions you have before I come up with some from my end. So the first one here is coming from Renia, so who is demonstrating who is expressing how interesting he finds that the cohort does not have a POE for the homozygous one. So he's asking, what do you think drives the AD phenotype in this cohort? A number of questions actually came from him, so I'll take that as the first. Please go ahead, Prof. Mahmoud.
All right, thank you very much for the question. So basically, the ApoE genotype that I showed in my presentation was not for these cohorts that we are currently genotyping. That was from the early iPSC lines that we generated. We have now a cohort of over 1,200 people. And what my group is doing right now is to genotype the entire cohort for ApoE so that we will know the genotype distribution. We are also sequencing the ApoE region so that we can know the haplotypes in the ApoE region. So I believe by the end of the year, if I get the opportunity to present elsewhere and you are there, I should be able to tell you the distribution of the ApoE genotype in this cohort.
That's interesting. And I'm wondering, because his other question is connected to having the numbers, which you have slightly addressed. But this is about the iPSCs that you have in the biobank currently. Do you have a number to that?
Yeah, so we already have about 11 iPSC lines generated. This is a laborious process, very expensive to generate one iPSC line. You spend at least about $3,000. So now we have about 900 biopsies from these individuals in the cohort. What we are interested in doing is we cannot generate 900 iPSC lines. I mean, we don't have the resource for that. But what we are doing is we are doing the GWAS, on the cohort, and we are doing ApoE targeted sequencing. Based on that data, when we see some interesting hits, some interesting haplotypes, some interesting kind of variants coming up, now we will now go back to the biobank to take the iPSC, the biopsies that we can now use to generate the iPSCs. So we hope to generate at least about 60 iPSC lines in the next 2-3 years. But if we have more funding, we want to be able to generate even more.
And speaking of funding, you can prepare for a little question in that regard, because that was something you didn't emphasize so much in your talk, as though everything came that easy. But before we go away from other questions from Remia, he also wants to know if the fibroblasts are stored and propagated in culture to enable future transdifferentiation experiments.
I would like to know who this Remia is, because the person has been paying attention and the person is asking all the relevant questions. Yes, this is exactly what we want to do. Because at the end of the day, if you think about it, if you have iPSC lines, iPSCs, when you are producing them, the epigenetic profile or the epigenetic kind of architecture gets reseated during conversion from fibroblast to iPSCs. And what that means is that the individual's life experience, the influence of environment, influence of other things, will all be lost or gone. And that means we cannot really be able to link this mechanism to the human, person if it is linked to epigenetics, if it is linked to environment or other things. We can only link that if it is linked to genetics. So because of that, with the biopsies that we have, we will be generating fibroblasts that future work would allow us to generate induced neurons. We already have a good working relationship with the pioneer of this, Andrew Yu at Washington State University, that allows for fibroblasts to be converted to induced neurons, which can now allow us to link the influence of environment and other factors to the cohort. So this is definitely part of future plans.
Okay, in the next few minutes, we may diverge to a couple more questions, maybe some having to do with how you achieved some of these feats. And on that note, we'll be taking the question from Dishala Bello, who wishes that you say something about how all this started, despite being based in the United Kingdom, and a little about what challenges you faced. and how scholars abroad can contribute to things like this. Remember, you have demonstrated a lot of first from the heart of Sahel. So how did you go about this?
This calls for an entire presentation of its own, to be honest. But
Yeah.
I think, in summary, I like to say two things. Where one thing that really allowed me to do this is knowing early on that I want to do this, do good science within my community, because I believe that our people are underrepresented and they are affected, and therefore I needed to do that. And so I had to start with outreach activities 13 or 14 years ago. Went to schools to inspire students. Royhaan was, has been an integral member of that team. So we ended up having a Pan-African outreach team through Trend in Africa, where I currently serve as an executive director, one of the co-directors. So we did this a lot, and that kind of slowly built onto doing our science festivals, establishing platforms like Science Communication Hub Nigeria, African Science Literacy Network, and that led to policy engagement. So basically, therefore, we started by taking baby steps that eventually allowed us to be able to engage diverse stakeholders, from the policymakers to academics, to now attract attention of the international and local funders, that now allowed us to have the confidence of the institution, of the local community, to say that we want to establish this, give us the required support, and for us to do that. The second thing which I really want to mention, I know a lot of people want to leave Africa because they see it as a problem. I
Mm.
see Africa as an opportunity, because we have the people that are genetically diverse, and that is a major strength that the African people and the African continent has, that no any other continent has. And most funders have now stopped funding people that will come to Africa to collect samples and go. Helicopter science, especially for Wellcome Trust, for example, and many funders, that has ceased to happen. So therefore, there is the opportunity from that angle for people who have the exposure to pursue this level of science to get funding. And secondly, we also have diverse fauna, flora, within the African continent. Now in Biotics, you have people working on agama lizard. We have people working on with giant cane rats. There are a lot of animal species that are unique and only available within the African continent, which also present another opportunity. So to be honest, when I now decided that I want to come back home, despite the fear, despite the scare, despite all the different things, I realized that actually I'm coming back to home, but also coming back to the land of opportunities. Okay, we've lost Dr. Royhaan. I don't know if there's another.
No, I'm right here. I'm right
Oh,
here. Thank you so much. So one thing we can add to that, of course, it's a whole presentation on its own. Neuros Sona is committed to both scientific and career development talks, and you have hinted on being ready to take baby steps and also exploring the available networks. You can all watch out for more talks on the journey so far in subsequent Sona webinar series, which we're hoping Prof. Mahmoud will yield our call to. Before we go, we're already on the top of the hour. Someone was wondering if you have gotten to the point, I think that's Ali Mali, was wondering if you have gotten to the point where you can say the effect of diet on some of these dementia-related diseases, whether in Yobe or elsewhere. Of course, you start with the UV samples.
So this is something that we hopefully will have an idea of as we analyze our data. So in the next 2-3 years, we should be able to hopefully understand if diet can influence that. But one thing that I should mention is there is a PhD student that is joining my team. She's at the University of Maiduguri. She's a microbiologist by training, and she's interested in understanding the relationship between the gut and the brain axis. And I believe that that kind of links up to the question. And I'm really excited to see what comes out of that. Should be working to identify how the microbiome in our cohort, influences, dementia. So again, I think there are a lot of exciting things that we look forward to from this cohort, and we are therefore interested in having people who want to come as fellows to work with me in the center, people who want to apply for funding. I'm happy to see how we can support your application to funders to come work with us.
Thank you so much, Prof. Mahmoud. Something always fascinating in your themes of talks has always been how you have gone from core science, neuroscience to now building platforms rather than just isolated projects. So that is something highly appreciated. We know that you are open for mentorship, you're open for support in all ways, like you have mentioned. For those of you who wish to know more about what goes on in BioRTC, Prof has demonstrated the website. Just visit the website and learn more about the opportunities, about the support available, about where you can fit in. Someone was asking about where a pharmacist will be. Do you have a role as a pharmacist in neuroscience or maybe in iPSC research? You can see much more about where you fit in. One thing perhaps which you didn't mention on the website, Prof, and also maybe now is also related to the question asked by someone. If another African university, you have been credited with being the first, you have done this so well, right? And so if another African university wanted to replicate what BioRTC has achieved, what would you refer to as the first 3 practical steps that you would recommend to them in this regard? Yes.
That's a tough one. Okay.
Don't mind me on behalf of Sonar community.
I think the first thing is for that institution to identify its area of strength. Who are the major pushers and shakers within the institution doing good science? Because what you want to do is to build something around people that can be able to sustain it. So assuming it's an institution X, and within institution X they have, department of anatomy, physiology, biochemistry, etc. And there's a commonality within this of scientists that are really, really good working in the area of neuroscience and everything. I think one of the first things is to convince them to build a common consortia of people with interest in a particular area. Secondly, I think they want to tap into now international networks. So if they have this group of people who perhaps have this track record or who have this interest, they can now easily reach out to consortia, to networks internationally. Because they are an institution, it will be easier for those networks to respond to them than individuals responding to them. I think it was tough for me, and I had to build this through years of work and also trained in Africa Digital. So institutions can easily do that. So if you now have that international network that has some alignment to a consortia that you have built, I think now the next thing is for that institution to also be honest with itself, to put in some funding to get some things going by either providing some level of infrastructure or some things that can allow the scientists to demonstrate to the network that they have linked them to that they are serious, they mean business. That can easily now allow them to access to attract international funding. It can easily allow them to be able to do more things. So for me, there are many different things they can do, but these are the 3 important steps that they have to do. 2 has to be them doing it by bringing or convening the people together, by providing a common central lab that can allow them to start doing something. And then the 3rd thing is to now give them the institutional backing by attracting networks or others that can allow them to continue.
Thank you so much, Prof. Mahmoud. There you have it, dear Sonar community. You need to know your strength within the local network, you explore the international network available, and convince your institution to provide the support where needed. And one thing you clearly added to that, maybe that started this whole thing, was also the commitment that you had as an individual. So if you're
Yeah.
trying this elsewhere, your commitment is what's going to be the fuel towards all of these steps that Prof has suggested. So as we
If I
draw
could,
today's webinar to a close, yes, Prof, go ahead.
if I could quickly mention, because that 3 steps is at
Yeah.
the institutional level. I know that a lot of people perhaps cannot speak to the head of department or a VC or whatever, and I don't want them to feel discouraged. You
Mm,
can see my example as also it can be driven by individual as well. And if you want to drive it as an individual, I believe also I will give you like 3 steps. One,
mm,
you know, get the qualification. If you have PhD already and you are highly, highly motivated, get the network as well. A network that believe in you and they can now be able to support your research direction. Again, Trend has been a major supporter of what I've been doing. That international network can now allow you to come back to your head of department or to your institution to say, look, this is my direction. And that international network can now be able to serve as your backer to say, okay, we were happy to support him with, let's say, equipment or with this and that, so that he can now be able to do the science. And that can also serve as your negotiating power, because if equipments are going to
or,
come, things will come, the institution can easily see
mm,
that maybe those things that you can attract can be beneficial to them as well. And that can give you some power
mm.
to be able to now convene a small lab that you can now eventually take forward.
In other words, you don't start by asking all the time. You have a lot to gain by showing what you bring forward. Thank you so much, Prof. That is clearly evident in the way you went about things. You demonstrated how much you are willing to benefit the community with, and that clearly has opened the governmental support that you have. It will be funny for you to realize that people keep sending questions, even some were just sent via email now. Someone asking what the roles microbiologists have. That's Kwame Sherima. Please, we are not able to take more questions beyond where we are today. You can always reach out, check out the website via RTC website, and you can always connect with Prof via that means. So as we draw today's webinar to a close, I'd like to express our sincere gratitude to Professor Mahmoud Bukar Maina for his outstanding presentation as always, and for sharing both his science and his vision with us. Of course, as regards the vision, you're still coming back. We hope you'll heed our call. So thank you also to everyone who has joined us from across Africa and beyond. We value your participation because it's what makes this webinar series truly continental as a platform, for scientific exchange. We appreciate you. Please keep an eye on SONA's communication channels for announcements of upcoming webinars, as well as our other engagements, including the forthcoming biennial SONA conference that we'll be holding along with the IBRO Congress in South Africa. And as we bring together outstanding speakers, as well as representing the breadth of diversity in African neuroscience. So allow me to conclude with a quotation from the Nobel laureate Rita Levi-Montalcini, who says, above all, don't fear difficult moments. And the best comes really from those difficult moments. We've understood how Prof. Mahmoud traversed that journey, the difficulties, the challenges, and really we're able to credit the heart of the Sahel with a lot of first from him and from his team. So thank you again for joining us. I believe you have all been scanning the code displayed, so you can give us your feedback, give your suggestions, recommendations of things you would like future talks to center about. And therefore, on behalf of SONA, please do have a wonderful day or evening, depending on where you're joining us from, and we look forward to welcoming you to our next webinar. Thank you all, and bye. Thank you, Prof. Mahmoud. See you some other time.
Bye.